Well, if you don't save our lives, you can't control our brains.
The idea of a bioterrorist attack is kind of the nightmare scenario because they're a pathogen with a high death rate would be picked. Now, the good news is, I'm not trying to depress you, it's tough enough
Too late.
right now.
Too late.
Most of the work we're going to do to be ready for pandemic 2, I call this pandemic 1, most of the work we'll do to be ready for that are also the things we need to do to minimize the threat of bioterrorism.
Well, Mr. Gates, thanks so much for being here. Thanks so much for the efforts you and your organization are doing to try to find the cure for this. And we'll see you again. Stay safe.
All right. Great time.
Give our best to everybody else in the bunker.
Thank you.
Life, do you hear me? Give my creation life!
The FDA has given the all clear to 2 pharmaceutical companies to begin a human gene editing trial. A joint clinical trial led by CRISPR Therapeutics and Vertex will use gene editing to target sickle cell disease and beta thalassemia. This will be the first confirmed CRISPR gene editing clinical trial to take place outside of China. According to a CRISPR Therapeutics news release, the gene editing therapy called CTX001 will be used to modify a patient's stem cells to increase hemoglobin levels in red blood cells. First trial of gene editing will be tested on 2 patients, with each being administered a single dose of CTX001. The patients will be monitored to see when the edited cells start producing mature blood cells, according to the news release. If everything goes well, the 2 companies will move to conduct clinical trials on a larger group of patients. CRISPR had previously been used by a Chinese scientist to create the world's first gene-edited babies.
Last year, Chinese scientist He Jiankui revealed he had successfully edited the CCR5 gene in the embryos of twin girls using CRISPR-Cas9 technology. By altering this gene, he expected the twins would be protected from contracting HIV. However, new studies show he might have inadvertently shortened their life expectancy as well. According to the study published in Nature Medicine, scientists analyzed the DNA of 400,000 volunteers, some of which had mutated CCR5 genes. Results show that individuals with non-working CCR5 were 21% less likely to live to age 76. Apart from increased mortality rates, results also show that altering the said gene could also make individuals more susceptible to influenza and the West Nile virus. The problem with mixing around genes is you never know what you're going to get. An MIT Technology Review report states that genes have many functions, so blocking one part of them may bring the intended results, but also unexpected outcomes. To the Dean of Harvard Medical School, George Daly, the experiment is an ethical and scientific failure. He told NPR, quote, "Even when we think we know something about a gene, we can always be surprised and even startled, like in this case, to find out that a gene we thought was protective may actually be a problem." To other experts, this is just part of the scientific process. Speaking to NPR, George Church, a professor of genetics at Harvard, said that new technological developments are expected to come with unforeseen consequences, adding, quote, "The first monoclonal antibodies were nearly deadly. The first gene therapies were indeed deadly. All kinds of means of transportation were and still are deadly. It's all about benefits versus risks." Hopefully, this study will serve as a wake-up call to stop messing with things we don't fully understand yet. Yeah, right. We're doomed.
one thing I can say, that we have to be humble. I've been doing infectious diseases now for almost 50 years, and I could tell you, I don't fully understand exactly what the mechanisms that is, and we really need to figure it out.